Bismillah, Assalamualaikum warahmatullah. You're listening to Microbiology lessons on microbial growth and control, Part 2. What you learn in Part 2 are firstly the physical factors that affect microbial growth, and secondly, how microorganisms interact with their environment. And then we'll look at how we can control microbial growth through physical antimicrobials. Factor is pH, which is how likely for the environment to donate hydrons to microorganisms. So it's the acidity of the environment. And then, of course, the opposite is also true, which is the alkalinity of the environment will also impact your microbial growth. We categorize our microorganisms based on their optimum pH range. Acidophiles usually grow optimally under pH 5.4. Neutrophiles prefer around, say, pH 5.5 to 8, and alkalophiles prefer once above pH 8. And similar categories go to temperature as well. If your microorganisms grows under 20 degrees Celsius, you can place it under psychrophile. If it does grow under 20 but can also grow up to 45 degrees Celsius, then it's a mesophile. Then above that, from about 40 to 70, we categorize them as thermophiles. There's a group of bacteria that can grow way above 70 degrees Celsius. We call them extreme thermophiles, and some papers will report them as hyperthermophiles. The highest one that I'm aware of is an archaea called G. barossi. What was it? Geogema barossi. So this barossi can reproduce at autoclave temperature. You know how crazy is that? At 121 degrees Celsius, where we use for autoclaving, where most microorganisms got burned and die, this barossi microbe is just chilling, wearing sunglasses and reproduce, having babies. Isn't that amazing? Another factor is oxygen exposure. One cool experiment to illustrate this is called shake tube inoculation. What you do is you melt nutrient agar, put it in a test tube. Then before the agar resolidifies, you put in bacteria. And then you roll the tube to spread the bacteria around. And then you incubate it. And then you observe the growth pattern the next day or the day after the next day, depending on your bacterial generation time. If the bacteria grow at the surface of your media, they are most likely obligate aerobes. So it means they can't live without oxygen. They need the oxygen as their final electron acceptor. And without oxygen, they die. Some bacteria are microaerophiles. They grow not on the surface, but slightly under it. These guys need oxygen, but not too much. So they can't grow at the very surface because there's too much oxygen up there. And then there's the facultative anaerobes. They grow all over the tube. They do need oxygen, and many of them prefer it. But God created them to be very adaptive in that if their environment lacks oxygen, they won't suffocate, they won't die. Instead, they switch to other metabolic pathways, anaerobic pathways that don't require oxygen molecules. And there are also bacteria that can't even stand having oxygen around. These are called obligate anaerobes. Not only that, they don't need oxygen, but oxygen effectively kills them. I remember in Grey's Anatomy, there were a couple of episodes where the hospital bought a hyperbaric chamber and the doctors did a surgery in it because the patient has sort of necrotizing fasciitis infection, which was caused by anaerobic bacteria. So they put the patient in the hyperbaric because it saturates the oxygen level, I think about 2 or 3 times the normal atmospheric level. The idea is you try to get as much oxygen as possible to poison the obligate anaerobes. And the reason oxygen is poisonous to obligate anaerobes is because oxygen metabolism produces free radicals like superoxide, and these anaerobes don't have enzymes to neutralize the free radicals. So enzymes like superoxide dismutase and catalase are crucial to fend off free radicals from taking out the electrons from the cells, from the cellular structural molecules. So without those enzymes, the obligate anaerobes can't survive when there is oxygen around. Like most life forms, microorganisms require water. So moisture content of the environment is also a microbial growth factor. So when the moisture level drops too low, microbial growth is either completely halted or decline. And I suppose spore formation becomes more likely because that's how they're going to try to survive. And for aquatic microorganisms, an additional factor is hydrostatic pressure. As you go deeper into water bodies like lakes or oceans, you're going to experience water pressing on you multiple times compared to the atmospheric pressure. So for many microbes, that pressure can crush them. But there are a group of microbes called barophiles. So these guys need that high pressure. If we take them to standard atmospheric pressure on land, they lose the 3D shape of their enzymes and they die. Another type of pressure is osmotic pressure. If your environment, for whatever reason, becomes hypertonic or hyperosmotic, water molecules will start leaving the microbial cells. So that leads to plasmolysis. If the environment is isotonic, you've got the balance between water going in and out. And if it is hypotonic, the microbes undergo turgidity, where the organelles start to swell up inside. Microorganisms that cannot thrive under high osmotic pressure, we call them non-halophiles. And the ones that can, we call them halophiles. Many staphylococci are halophiles. And then if they really need osmotic pressure, like certain halobacterium, we call them extreme halophiles. And lastly, radiation. The reason radiation level can affect microbial growth is because the energy beam can mess up the DNA structure, and that may introduce mutations that would be inhibitive to reproduction. So they can't grow because their DNA can't be translated into proteins. There's one species that's quite popular, radiodurans. So that radiodurans bacteria can withstand radiation, I think, 2,000 times the level that kills us humans. So those are the half a dozen environmental factors affecting microorganisms. Now, being living microorganisms, microbes aren't just passive entities to their environment. They interact with their environment. One key example to illustrate this is quorum sensing. In quorum sensing, cells interact with one another, and that interactions affect their phenotypes as a collective. What that means is, if you have isolated cells, they behave one way. When those cells are in a community, they behave another way. The way they get into a community is either they grow there or they are free-swimming cells that land on existing community. Once they land on the site, they start producing a matrix of chemicals that help them bind to the surface. Think of it like an alien colony ship arriving on a new planet. So once they land, they establish a colony site. They build structure. In the microbial case, we call that structural matrix. It helps stabilize their position so that they're not going to be washed away by the environment. In the beginning, the new colony is small. So when they release signaling molecules, not much happens, meaning you can't see significant change in their phenotypes. Over time, as the biofilm colony expands in number, the signaling molecules increase. As the concentration of signaling molecules increase, they get to a level where the signals trigger gene expressions. So that level is called quorum. So when the signal magnitude is under this level, the genes are not triggered. And once you achieve the quorum, the genes are triggered for expression. And those gene expressions lead to protein expressions, specifically the enzyme proteins. And this can be any types of enzymes depending on the signaling molecules being released. For example, they could be enzymes related to food digestions. So you see, as isolated cells, the bacteria might not be able to break certain type of food particle. But as a biofilm, as a collective, because they achieve the quorum, they now can use the food particles. So that's a new phenotype. So to summarize that one, if you have isolated cells, they behave one way. When those cells are in a community, when they achieve the quorum, they behave another way. So that's the phenomenon we call quorum sensing. Another way microorganisms react to the environment is sporulation. So there are at least two types of spore formation. One is among fungi, especially, where they form spores to reproduce. The second type is by certain bacteria, where they form spore as survival mechanism. And here we're talking about the second one, sporulation for survival. Bacteria don't do this for reproduction. For reproduction, they stay in vegetative state. In their vegetative state, they reproduce through binary fission. I talked about this process in the previous episode, so check that one out again if you forget. So bacteria stay in that vegetative state when the environment supports their reproduction. When that is no longer the case, when there is an environmental stress that threatens the bacterial survival, they switch into sporulation mode. So it begins with the formation of axial nucleoid. What that means is the chromosomes and the nucleoid starts to replicate into a line of a single geometric axis, a single long line. And along that axial nucleoid, you're going to see formation of a core somewhere. I said somewhere because for some bacteria, the core is formed at the edge of the nucleoid, and for other bacteria, the core is formed in the middle of the nucleoid. So this core is then start to be surrounded by endospore septum. In this septum, by the way, it's not just chromosomes. You'll detect some proteins and some elements like calcium ions. So basically, the core has all the supply that they need to survive. The septum then form two layers or double membrane. Both layers put peptidoglycan in between them. So that peptidoglycan overlay is called cortex. Why does it make this? We're not 100% sure, but we think because the cortex protects the spore from extreme osmotic changes, to avoid plasmolysis. So now you have cortex surrounding your spore, and then the mother cells put carotenoid protein coat around the cortex. That protein coat is the armor. It protects the spore from hostile elements outside. And that's the last thing the mother does. She gives that protection and then she starts to die, to lyse, leaving you with protein-coated spore. And that spore can survive and sleep for years and years. When the environment becomes favorable again, the spore wakes up. You'll see the coat starting to break down. So you can detect most peptidoglycan fragments at this stage. And eventually the coat goes away and the cortex turns back into the normal membrane and the cell arises again as vegetative cell. So that's your story about sporulation. Alright, now that you know a bit about how microorganisms grow, we can talk about how we can exert some control over microbial growth. In controlling microbial growth, we can take a physical approach or chemical approach. In physical antimicrobial theory, you've got a couple of basic concepts. First is sterilization. So this is when you kill all microorganisms, all of them. And that concept can be contrasted with disinfection. In disinfection, your goal is not to kill all microorganisms. Rather, you want to kill enough of them so they no longer pose pathogenic threats. Enough so that you don't get sick. So that's the difference between the two. Another comparison that you can make is between antiseptics and disinfectants. So antiseptics are what you use on living tissues to kill pathogenic microorganisms. Disinfectants are what you use on non-living objects. So in popular conversation sometimes, social media, you see these two terms are used interchangeably. But in professional writings, in professional conversations, they are technically different concepts. So just be aware of that. All right, now let's talk about some physical approaches to control microbial growth. When you hear me talking about each of these, I want you to reflect and relate it to the microbial growth factors that we discussed earlier, that will deepen your thinking and help you remember them. Number one is high temperature exposure. The reason intense heat kills microorganisms should be obvious to you by now, but there are certain technical terms that you want to explore later after you listen to this audio, terms related to heat treatment. First is thermal death point. It's the temperature that kills all the bacteria in a 24-hour-old broth culture at neutral pH in 10 minutes. Second is thermal death time. So this is the time taken to eliminate 100% of the bacteria at a specified temperature. So say for instance, your thermal death time is 10 minutes. So that simply means that 10 minutes is how long you need to kill 100% of the bacteria. The third one is decimal reduction time. So instead of how long you need to kill 100% of the bacteria, the decimal reduction time tells you the time taken to kill 90% of the bacteria. Now you might ask yourself, why do we differentiate between 100% killing time and 90% killing time? This distinction is important in food microbiology because you see, in medical microbiology, for a surgery, for example, if you want to sterilize a forceps, you don't need this technical distinction. You just kill the 100% of bacteria. But for food, you want to balance between killing enough bacteria to make sure your consumers don't get ill and at the same time, not compromising the flavor of your food. Basically, you want the food to be heated just long enough to be safe to eat, but still taste good. Heat treatment can be of two types, dry and moist. For dry heat, in the laboratory context, we use oven. For example, you can set up your oven to be just above 70 degrees Celsius for 1 hour, and that's enough usually to sterilize your materials. Moist heat, we use autoclaves. So you heat your stuff using steam under pressure to get to 121 degrees Celsius. The sterilizing itself just takes around 15 minutes, but the autoclave machines typically need to run much longer to ramp up to 121 and then to go back down to room temperature before you can open the door safely. Another method is pasteurization. So this is most commonly used for milk. You have flash method, which is 71.6 degrees Celsius for 15 seconds, and holding method around 63 degrees Celsius for half an hour. And don't drink, if you have the pleasure of visiting a farm, don't drink fresh milk from cows that has not been pasteurized. Even if it looks clean and organic and stuff, you're going to get sick. It's been documented scientifically. And if you have seen UHT on the milk boxes, it means ultra high temperature. So the milk was treated by raising the temperature from 74 to 140 degrees Celsius and dropped back to 74. And all of this in less than 5 seconds. Subhanallah, amazing technology. So when your goal is inhibition rather than complete elimination, you can use the low temperature treatment as an option. Refrigeration is an amazing technology that we often take for granted. Now, one thing to remember at home, if you recall, even at low temperature, there are still microorganisms that can thrive quite well. Remember the psychrophiles? So make sure you regularly clean the fridge. So help your mom, keep your family from getting sick. Freezing is another option for longer storage. It causes most of the microorganisms in your food to be inactive. When you work in a lab later, you're going to find freezers that go down to minus 80 degrees Celsius to store your stock samples. So it's different from your typical home freezers, which just go only to negative 20. A more low-tech approach for controlling microbial growth is drying your food out. So this works really well because you basically take out water molecules, which is required in most enzymatic pathways. And a traditional way of doing this is just a direct exposure to the sun, which I saw my grandparents used to do. They dried fish and prawns. So on top of dehydration, that direct sun exposure also helps kill many of the microorganisms. Another method of drying is freeze drying or lyophilization. You use machines to quickly freeze your sample, and then you use a vacuum system to suck out all ice particles from your sample. Like when they make instant coffee powder, this is the method they use. The third approach is using various forms of radiation. UV is common, useful for viruses, less so on bacteria, I think, because often they've got sophisticated DNA repair system. Ionizing radiation has a much shorter wavelength. What it does is it ionizes or kick electrons out of the orbits of atoms, so turning those atoms into ions. So it has been used by many developed countries to sterilize food. Microwave radiation. This one you use at home, so it doesn't kill bacteria by damaging their nucleic acid structure like UV or ionizing radiation. Microwave simply agitates water molecules, which then releases heat. So it's the heat that kills the microorganisms, not the wave itself. For visible light spectrum, I kind of mentioned it indirectly when I talked about drying just now. So direct sunlight is bactericidal. It kills bacteria. So at home or in your college room, it's a good idea to open your curtains and open your windows, by the way, because glass can actually block certain wavelengths. So do this to kill harmful bacteria in your room. Beside light waves, you can also use sound waves as well, such as cavitation or sonication. In cavitation, you expose your sample to ultrasonic waves over 15,000 hertz. So that causes the formation of vacuum in the cytoplasmic fluid, and those vacuum pockets will damage the structural integrity of the cells. So basically think of it like this. Cavitation make vacuum holes in your cells, and because of the holes, the cells break apart and die. For sonication, you can use sonication to kill bacteria as well, although I don't think it's the first choice in most conditions in industry. But it does work. It uses sound waves to vibrate your bacteria to death. Sometimes in the lab, you can't apply heat or radiation to your sample because you want to preserve proteins within your sample. So one option to decontaminate your sample is by using these expensive filters. It's actually really hard to push your sample through them sometimes. So what you can do is you use the larger pore size first, and then you use the smaller filters. It's nothing is complicated here. It works just like your kitchen coffee filter. So it blocks the larger microorganisms that you don't want in your sample. And lastly, osmotic pressure. Osmotic pressure treatment is common. It's just we don't call it that in popular discourse. If you have had kimchi or other pickled dishes, those food are treated with osmotic pressure. We use high level of salt or sugar to make the environment hyposmotic or hypertonic for microorganisms. So they can't survive it because of plasmolysis. So think of our lesson here the next time you have your kimchi. Yeah? All right, there you have it. The next episode should be shorter, inshallah. I pray that you benefit from this lesson. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.